And here are some things that I want you to think about. I'm going to go relatively quickly through some of this stuff, but I think this is quite honestly of everything that we do in this class. This is probably the most important thing, because this will determine how much you learn, which is what's really important to me. And for you, this is what's going to determine things like how much credit you get in the class and what kind of a grade you get. And some of you are really interested in learning, which is great. Those of you who are interested in learning, almost all of you are interested in learning, or in credit and grades as well. And some of you really are only interested in credit and grades, and that's okay. It's not, my job is to try to convince you that it's worth being interested in learning stuff. My job is not to make you feel bad if you're not interested about things, okay? So here's what I want you to imagine, and this is those of you who, sometimes you have to speak sharply to the technology. So those of you who have made graphs in your math class and you've been like, when are we ever going to use this? Here's an example. This is a great way to use graphs to illustrate complicated ideas. And in this case, we're going to be thinking about two things. One thing that we're going to think about is the degree of difficulty involved in learning, in material, like academic concepts. So if we think about some sort of a concept like cellular organelles or evolution or how lichens work or whatever, we can look at it in a real simple way. And we can look at it in a really complicated way. And what most of us would find is we already know the simple stuff. And the really complicated stuff might be out of our league. So if we were to evaluate, kind of like one way to think about it would be interest, right? And so one way to think, and I'm going to kind of invert this for a reason. One way to think about this is things that are really interesting are down here, and things that are incredibly boring are up here. And what you would find is that if you think about things that you already understand, and you only think about the things about them that are really, really simple, it's pretty boring. Like if I were to stand here and spend 2 hours telling you that plants are like things that grow and they have green leaves on them and they have stems and trunks and their like roots are in the soil, like it just would be a snooze fest because you already know all that. It's not that interesting to you. So that would be something that would be highly boring. But as we got into things that maybe you don't know so much, like maybe I would talk about things like, well, how many of you know where most of the mass in a plant comes from? Does it get it from the soil? Or does it get it from sunlight? Is it from water? Like where does the plant come from? Does anybody know? What makes the leaves in the stem? Where does it get it? Like you start with a little baby plant and it grows for a year and now it's a bigger plant. Where did all that stuff come from? Does anybody know? Colton, do you know? You probably would, if I told you where it came from, it would probably be within your ability to understand it, right? What do you think? Angel, do you know? No. Ava, do you have any idea? Where does the stuff in a plant come from?
The seed.
The seed. But it starts this big. So that starts it off, right? But how does a seed, if this is where it comes from to begin with, how does it get bigger? How do you start with something really, really small and have it get bigger and bigger?
Nutrition.
What's that?
Nutrition.
Nutrition, right? It has something to do with, there's an input, right? Where does that come from? The soil? Water? What do you think, Georgia?
The sun.
The sun?
Nice, Georgia.
Yeah. All those things are great ideas. Here's the answer, by the way. It comes from the air. Most of the mass in the plant comes from air. The plant takes carbon dioxide from the air and it chemically alters it using energy, which has no mass, from the sun, and puts it together to make plant stuff. That's where it comes from. So you have a plant that weighs half a pound and then a year later it weighs 2 pounds. That plant has absorbed very close to 2 pounds of carbon dioxide from the atmosphere. That's where it comes from. So you could understand it, right? But here's another question. How does that happen? How do plants take energy from the sun and use it to pull carbon dioxide out of the air? Like now we're starting to get to a place where it's not easy to understand. It's still kind of cool to think about though. Like on some level we can pull this together. On some level we have some conceptual ability to understand that plants are like pulling carbon dioxide from the air and hooking them together, right? So that brings us down to here, where there's a place where it's not so simple that we're bored, it's complicated enough that we can understand it and we're interested. Now here's the problem, is that then if we started talking about, say, electron orbital levels and valence electrons and energy balance, and if I asked you to memorize the Krebs cycle and count the amount of ATP that happened at each turn of the Krebs cycle, you would legitimately want to like throw something at me, because that would be incredibly complicated, way over your heads. You don't have the chemistry and you don't have the physics to understand that yet. So if we think about stuff that's too complicated, then it's boring, not because you've done it already, but because it's boring to talk about things that you don't understand. And you can kind of picture where I'm gonna go from here. There's a sweet spot right here, right in the intersection between the things that you don't know yet, but you can understand them, the things that are like so easy that you don't want to talk about them anymore, and the things that are so complicated that you can't get them. This right here is where we want to land. This is what I think of as the sweet spot for learning. And here's what makes my job really challenging, and it's kind of makes your job challenging too. This is different for everyone. Some of you understand a little bit about photosynthesis. Some of you know the equation for photosynthesis. Some of you understand that light comes in photons, and photons are energy, and that the pigments in chlorophyll in the chloroplast of the plant can absorb that energy and like reroute it. Some of you, that was nonsense. Some of you understand that plant roots pull water and micronutrients from the soil, and the leaves get carbon dioxide from the air. Some of you don't realize that. Some of you don't understand that plants both respire and respire. The respiration is the opposite of photosynthesis. They photosynthesize, they respire, and some of you really don't even understand like what chlorophyll is. You've never maybe heard of it, or you don't remember, and so what's difficult for somebody might be really easy for somebody else, and vice versa. So part of your job on a daily basis in this class is to figure out this spot for you. It's different for everybody. What I want to do is set you up so that you can find that spot. When I ask you to think about cellular organelles, for example, which is what we're working on, I don't mean memorize the definition of a cellular organelle. I also don't mean tell me down to the last electron how these cellular organelles work biochemically. What I mean is figure out what you already know about these organelles and kind of skip that. Find the stuff that's interesting to you and avoid the stuff that's too complicated for you to understand. It's different for everybody. My son is in 5th grade, and he got in the car the other day and he was like, "Oh, Dad, I have to give a speech on bricks." And like, really, is there anything more boring than a brick? Can anybody here think of anything more boring than a brick? Yeah, I can too. Yeah, but I mean, so think about this for a second though. Like, you're a 5th grade kid, and you're asked to do a project on a speech on bricks, and that to me, as a 5th grader, if you would have said, "Here you go, pal. Here's your project." Of course, I have bricks in my classroom. I mean, come on. Like, those of you who don't know me are probably not surprised. Those of you who do know me knew this was coming. So here we've got this brick. Oh man, it's a yawner. Except here's a couple things that are kind of interesting about it. What is it made of? So what is that? My son needs to do a report on how bricks were made in early colonial America. So right now, if you told me, Regar, go make a brick, I would go to a hardware store, I'd buy a bag of cement, and I would like mix it with some water according to the instructions, and I don't know, I would get a little like box like this, like fill the box up, and then like let it harden for a while, and then I'd have a brick. You're not gonna believe this, but they did not have Home Depot in 1684.
Wow.
Okay, it's sort of a new thing. You didn't go to the store and buy concrete. And if you were a settler, a colonial settler from Europe that was in what will be the United States of America in 1684, there aren't even like ready sources of these materials. Like nobody knows where any of this stuff is. How do you make a brick? If you are a 25-year-old, like hale and healthy and hearty individual in 1684, and you don't have Home Depot, and you don't have cardboard boxes, and you don't have an instruction manual, how do you make a brick? Does anybody in here know? Would anyone know? What would you do?
Yeah, I guess I'd say find clay and then like you could make like imprints in the ground.
So you'd like find some clay? Is that what bricks are made of?
Might have been.
Just plain clay? Like in 1684, did they make bricks from just plain clay? Were they just like pottery blocks? I don't think so. I mean, we have bricks. Like there are buildings that were built with bricks from like several hundred years ago. It's not clay. That's not what bricks are. I mean, you can make clay bricks, but that's not what bricks are. Yeah.
Isn't it like hay and mud?
Hay and mud.
That's a
You can make buildings from hay and mud, but I don't think that brick as a building material was just hay and mud. You can do that. Like you can make all sorts of structures, but if what you wanted was bricks, then you wouldn't have just like gotten clay and heated up, and you wouldn't have used like hay and mud. What does anybody know what it's made of? So here's where it gets really interesting. Right now, we started with, oh my gosh, like I have to give a 1-minute speech on bricks. What could be more boring? But now, at least a third of you are really interested in what's going on. And here's the thing about it that I love. I mean, this is one of the things I love about this job more than anything. I'm super curious too. I don't know how they made bricks in 1684. I don't know any of this. Maybe it is just clay. Maybe it's hay and mud. I do not know. I don't know how they did it. And the best thing about all of this is I really want to know now. So when my son and I were talking about this, we kind of went in a different direction. We were talking about like the heat of the kilns that they use and trying to figure out, well, you mean, how do you measure temperature? Like you don't go to the store and buy a thermometer. Like if you're a brick maker in 1684, you're not like, hey, do you have any thermometers for sale? Like, where did you get your thermometer? How did you know what temperature anything was? How did you make a kiln? How did you get it to the right temperature? How did you keep your bricks from exploding? There's different kinds of clay, if they're made entirely of clay. How did you know whether to use that clay or this clay or that clay? How did you even know whether the soil you were using had enough clay in it to be used as a brick?
Trial and error.
So trial and error would be a big one, right? Yeah, you'd have to. You'd have to use trial and error. But you'd also have some knowledge to start with. You'd have enough information that your pool of trials would be somewhat limited. Yeah.
And wouldn't there be like some people around who know something about bricks, so you could ask around if
You're the person. That's you. If you're the brick maker, you know more than the other people. If you're a brick maker who just got off a boat in Northern Virginia in 1684, there may be nobody else, right? Like Native Americans lived there and they knew everything about everything they did, but they didn't make bricks. So they could find food, they kept the colonial settlers alive, but they weren't the ones who were like, oh, here's how you make a kiln. Yeah.
But maybe Native Americans do work to find clay.
That's right. So they would have seen, like the Native Americans did have pottery. So they would have said, hey, where'd you get the stuff to make that? Right? Exactly. That's exactly the kind of stuff you do. So the point is, no matter what you're looking at, you can find the place where it's of interest. We can find this spot, whether it's for cellular organelles, whether it's for bricks, whether it's for things that in general you tend to gravitate to, which might be music or sports or video games or art or whatever it is that interests you. You find this place naturally when you're on your own. Like if you are given an afternoon and you've got like whatever your like favorite hobbies are, art or music or whatever, you've got your stuff, you go to this place automatically. How many of you are learning to play an instrument right now? It's kind of fun, right? To like, you find you try something new. You don't try to do something that you can't do, because that would just be frustrating. Sometimes it's kind of fun to do the easy stuff, but usually you try to do something that's a little bit more. It's kind of fun to learn. And that's where I want you guys to get today. The problem with this is, since you're all in different places, I can't just say, do this, do that, do this. What I like is to teach you how to find this spot yourself. You already know how to do it outside of school. And many of you are going to be able to transfer that skill to this classroom pretty easily. But some of you are going to struggle, and that means you need my help. So that means you're going to come to me and you're going to be like, Brigar, I got that the endoplasmic reticulum is an extension of the nuclear envelope. I get that it plays a role in transporting proteins. And I see that rough ER is studded with ribosomes that make proteins. Can you help me figure out, that all makes sense to me, but I'm not seeing a mystery here. Okay. I'll help you find the mystery. I'll help you determine the mystery. Like, for example, most of you probably don't remember this, but if you look at the endoplasmic reticulum in cross-section, it looks like this. So here's what the nucleus of the cell looks like. It's kind of round usually, and it's made out of basically the same stuff that the outside of the cell is. So the outside of the cell would be here, and here's the nucleus, and then the endoplasmic reticulum comes off, and it does this. And my question to you is, why? And some of it has ribosomes on it. You might not remember what ribosomes do. They make proteins. And that's the rough ER. And then there's smooth ER. Why does it look like this? What is the advantage of this shape? How do you get this shape? Why?
I don't know, but I have a different question.
Okay, go for it.
What's the most important cell in our body?
The most important cell? Kind of cell? Depends. It depends on your definition of important. We know that people can be alive with only brain cells functioning really. The few other ones. But if you want to be able to interact with other people, you need more than that. If you want joy in your life, it's more than just your brain. You need other things. We crave that. We're human. There is no one most important cell philosophically, but you could certainly look at each cell and look at the role that it plays. There are certain cells that if we said if we have a human being without brain cells, functionally that human being is not alive. We know that. We can keep the rest of the cells alive, but that would not be what we would call a living human. Yeah.
How advanced does your brain have to be to make compromises or negotiate?
We can talk about this more later, and I want to. But those are questions I can't really answer. They're interesting, and I like to think about them. But they're more like musings and ponderings. There's no one answer to those. My question about what is the advantage of this weird squiggly shape, this is something you could find out. This is something you could look up. This is something that you could understand. And you could think about it with a little bit of mathematics even. And so this is the place where if you come to me and you're like, I get the whole endoplasmic reticulum thing. I know what it does, I know where it is, I know what it's made of. I'm gonna ask you, okay, why is it shaped like this? And you'll go figure that out. And then you'll come back and you'll be like, okay, I get it. I know why it's shaped like that. And then I'm gonna ask you some other question. So I'm gonna ask you, for example, Why is the rough ER closer to the nucleus and the smooth ER farther? That's a pattern that you'll see. Why is that? There's an answer to that, and it's all like mysteries. So that's one thing I want you to think about. And this really is where I want to guide your work today. I want you to be invested in learning about stuff. I want it to be interesting to you, because if it's not, you won't really learn much. So find this place. The second thing that I want to tell you has to do with this is not part of our learning objectives, but this is always in the back of my mind, and I think this is super cool. So it's kind of a process of discovery that we have short-circuited. What we start with in this class, when it comes to like cellular biology, cell biology, molecular biology, we know all this stuff. We know what DNA is, we know what it's made out of, we know what it looks like. We know where organelles are, we know why they're there. We can kind of piece together all this stuff. But here's what I want you to imagine. In the 1700s, when people were first developing light microscopes that were of really good quality, they looked at cells under the microscope. They didn't even know what they were. Like, when people first saw cells, they were like, I don't know what that is. They called it a cell because it kind of looked like cells in like a prison or like a beehive. So they were just like, well, when I look through this microscope, I see these things. They looked at cork, was one of the first things that they looked at. It has pretty big cells, so it was easy to see the cells. So this was like step one. They see the cells. They don't know what they are. Then they start looking at more living organisms and they see more cells. And they're like, aha, living things are made out of cells. And then they get their microscopes better and they can look at one cell kind of close up. And they start to be able to see some stuff, like with the cork, they might see something that sort of looks like this, and they realize, oh wow, the cell has two layers. It's got an outer layer and an inner layer. The outer layer has kind of sharp edges, and the inner layer is a little blobby. So they start to put some things together. They call this the cell wall, because it looks like a wall. They call this the membrane, because it looks like, like they didn't have plastic. They didn't have plastic bags. We would have said it looks like a plastic bag. You know what they probably thought? I'm guessing. They probably thought it looked like an animal bladder, because that's what they used. Like, people would take pig bladders and cow bladders and use them the way that we use plastic bags. They're awesome for that. So they were probably like, well, it kind of looks like a bladder. That's probably what they were sort of thinking. They called it cell membrane. And then they figured out, oh hey, if I dump this like goo on a cell, then some of the cell ends up absorbing the color of that goo. So if I have some green goo, and then I look at the cell, then I see green spots. And they had no idea what these things were, but they started to see some patterns, like, well, some of the spots are bigger than others, and some of the spots have patterns in them. And they had no idea what these things were. They gave them names because they wanted to be able to refer to them, but they didn't know what they did. And here's where it gets super interesting for me, which is, okay, what next? How did they figure out, when you look at the diagram from Cells Alive, all those details, how in the world did they do that? When you were looking through the microscopes the other day, did it look like Cells Alive? How did they figure out what these things did? How do you figure out what ribosomes do? You can't harvest them, we can't see them with our naked eye. You can see them in a microscope, maybe on endoplasmic reticulum, but how do you get them off? How do you figure out what these things do? All the stuff that you're going to be doing today, all of it, had to be figured out by people who had no idea. And to me, that's really fascinating. When I think about this stuff, you start working on the Golgi apparatus, which is protein alteration and shipping, you might ask yourself, how in the world did we ever figure out what this thing did? That is a really cool question. It's not critical for what we're doing, but it's critical for driving your interest, which is what I really want. Yeah?
So with the cork, with the cells they would be looking at would just be some cells from a tree?
Yeah. They just put stuff under microscopes. They did the same thing that you all would do. if and when, like when I brought microscopes in, like I saw you, a whole bunch of you just stuck your fingers under there. Good. That's how people learn stuff. Everybody should do that, right? Like that's what, you did that 10 years ago when you were kids. If you had a microscope in front of you, you would have been like, okay, well I'm gonna put my finger under it, because that's what you do, right? You look at stuff. That is what learning is about. That's what science is about. That's what it should all be about. And that's where I want to get you guys back to. So my goal is to get you guys back to thinking about things the way that you did when you were 5. You were a lot smarter when you were 5 before we like dumbified you. So here is the goal for you today. There's a lot. I'm going to ask you to do a whole bunch. I'm going to ask you to work your tails off over the next 56 minutes of your life. In the next 56 minutes, I want you to first come up with analogies for each of the cellular structures that are listed in the Cell is a City poster assignment. I have an example here, if that's useful to you. I'm not going to go through it with everybody, but I'm happy to work with you individually if you would like help in doing that. The second thing that I want you to do is after you've come up with good analogies, I want you to just run it by me. Like before you do anything else, get my opinion on the work that you've done so far. And the third thing that I want you to do is I want you to get it all on one of those big whiteboards, and we'll take a picture of it at the end of class. And what should show up on the whiteboard is going to look kind of like this. Hang on, this is old, so I got it here. It's going to look kind of like this. So this was just an example that I did a few years ago, actually. So if this is my shopping mall, and I wanted to have an analogy for a mitochondria. What a mitochondria does is it takes chemical energy stored in glucose molecules, and it processes them in such a way as to produce chemical energy in the form of something different called ATP. One form of energy goes in, a different form of energy goes out. At a shopping mall, that happens maybe, there's probably like a power room. But we have one here. Big buildings have like power rooms. And in those power rooms, they have a whole bunch of breakers, like circuit breakers and like panels. And what comes into those panels is really high voltage. It's generally 240 volts coming in. What goes out of those panels is 120 volts. That's what these plugs all are. The panel takes one form of power and it converts it into another form of power. The power room of a shopping mall is like mitochondria because it takes one form of power, one form of energy, and converts it into a more usable form. Many of the things that we plug into these outlets would simply explode if we plugged them into a 240 volt outlet. Not everything. These are designed to work with both. You could use this in Europe, for example, where they run on 240 volts. But a lot of things wouldn't. If you took a regular light bulb and plugged it into 240 volts and turned it on, it would just go pop, and that would be that. That would be the end of the light bulb. So that's what a power room at a shopping mall does, just like that's what a mitochondria does. This is what your whiteboard is going to look like, only you're going to have something like this for every one of those cellular organelles and structures. And it's not going to be analogous to a shopping mall. It's going to be analogous to an entire city. So Lucius, hold on. I'm going to get your question in just a second. Before I do that, I want to make sure that everybody understands the basic parameters here, which is that you can work in pairs, and you should work in pairs. We have enough whiteboards for people to work in pairs, but not in groups of 3 or more. If you want to work individually, then we may not have a whiteboard for you, so that's fine if you want to work individually, but then you're going to just work on paper, and that's totally okay. It still needs to follow the basic criteria, which are, hold on, got it here, which are that you need a basic image showing each organelle as part of a city. You need descriptions for each image telling what it does in the city and how that's like the corresponding cell structure. You need to make that analogy clear. It needs to be explicit. This you can't read, but it says, power room of a shopping mall is like a mitochondria because That is an explicit analogy. And then the last thing, I was kind of like being a little jokey here, make it lovely. I'm not the world's greatest artist. But, I mean, if I can draw something that looks like this, then you can draw something that looks 12 times better than this. I did this in 30 seconds 5 years ago. You can do better, all right? So that's your job. This is due at the end of the period. Your grade is gonna be determined entirely by what happens in your brain. I'm gonna be watching what you do and interacting with you and grading you based on what you're thinking about, not what ends up on the paper. If you're thinking about the right stuff, your whiteboard or your paper is gonna look awesome and you'll get full credit, all right? If you have questions, let me know.